Good morning, students. We have studied the glycolysis cycle of the common pathway of respiration. So, in that carbon pathway, we have seen that the respiration process completes in three steps. One is the glycolysis, which takes place in the cytoplasm. The other one is the Krebs cycle, which takes place in the mitochondria, and the third one is oxidative phosphorylation, that also takes place in the mitochondria. So glycolysis end product we have obtained, that is the pyruvate, and this pyruvate enters into mitochondrial matrix from the cytoplasm. So pyruvate is synthesized in the cytoplasm by the process of glycolysis. Then this pyruvate enters into mitochondrial matrix. So if suppose this is the mitochondria, so this will be the mitochondrial matrix, and this will be the cytoplasm. In the cytoplasm, glycolysis will take place, in which glucose is converted to pyruvate, and this pyruvate enters into mitochondrial matrix, and in this mitochondrial matrix the TCA cycle, or we also call it Krebs cycle, takes place. So today we will be studying about the TCA cycle or Krebs cycle. So first of all, the pyruvate or pyruvic acid, this pyruvic acid, or we also call it pyruvate, which was the 3 carbon compound, we got 2 molecules here with 1 molecule of glucose. So 1 molecule of glucose broken into 2 molecules of pyruvate. Glucose was 6 carbon compound, and it will break up into 2 molecules of 3 carbon compound. So pyruvic acid is a 3 carbon compound, and we have got 2 molecules. So this pyruvic acid will require coenzyme A to combine with acetyl coenzyme A, which is a 2 carbon compound, by releasing carbon dioxide and by getting reduced by NAD to NADH plus H. So then this carbon acetyl coenzyme A will get hydrolyzed by addition of water, and it will combine with oxaloacetic acid, which is a 4 carbon compound. So this acetyl coenzyme A, 2 carbon compound, oxaloacetic acid, 4 carbon compound. Then the condensation reaction will take place. So condensing enzyme, that is citrate synthetase, will act here and this reaction will take place in the presence of pyruvate dehydrogenase. So as it is a dehydrogenase enzyme, so this will be removing the hydrogen ions from pyruvic acid and a molecule of carbon dioxide, forming the acetyl coenzyme A, that is the 2 carbon. So this was 3 carbon, 1 carbon is lost in the formation of carbon dioxide, so it will become 2 carbon compound, and then in the presence of water molecule it will combine with acetyl coenzyme A, that is the 2 carbon compound, it will combine with 4 carbon compound, and it will become citric acid or citrate, which is the 6 carbon compound. So 2 carbon from here, 4 carbon from here, it will make the 6 carbon compound. Again we'll get 2 molecules here. So first reaction we can say this one, second reaction we can say this one, then next there will be the removal of water molecule in the presence of enzyme aconitase, and it will form cis aconitic acid. This is also 6 carbon compound. Then the third step is, again there will be the addition of water molecule in the presence of enzyme aconitase. It will become the isomer isocitric acid, and this reaction is the isomerization reaction. So cis aconitic acid will get converted into its isomer, that is the isocitric acid. Then this is also 6 carbon compound. Then from this there will be again dehydrogenation. NAD plus will receive these hydrogen ions and it will become NAD plus, NADH plus H plus. In the presence of enzyme isocitric dehydrogenase. Again dehydrogenase enzyme means removal of hydrogen ions from this. It will become oxalosuccinic acid, again 6 carbon compound. So this is the fourth reaction. Now this will be the fifth reaction. Oxalosuccinic acid will become alpha ketoglutaric acid. This is the 5 carbon compound. So again there is loss of 1 carbon, so there will be removal of carbon dioxide in the presence of enzyme oxalosuccinic decarboxylase. Then alpha ketoglutaric acid in the presence of enzyme ketoglutaric dehydrogenase in the presence of coenzyme A, it will release CO2, and again there will be the removal of hydrogen ions which will be accepted by NADH. So this is the sixth step. It will, after losing the carbon dioxide and the hydrogen ions, it will become succinyl coenzyme A, which is the 4-carbon compound. Because it was 5 carbon, it lost 1 carbon in carbon dioxide, so it will become 4-carbon compound. Then it will lose the coenzyme A, and by addition of water it will become succinic acid, again 4 carbon, and there will be the synthesis of GTP, that is the energy molecule, and ADP will get converted into ATP, and GDP will get converted into GTP in the presence of enzyme succinyl thiokinase. Then succinic acid will get converted into fumaric acid. Again it is 4-carbon compound, and in this reaction FAD will get reduced to FADH2 in the presence of enzyme succinic dehydrogenase. So this was 6th reaction. This is the 7th reaction. This is the 8th reaction. Then from fumaric acid there will be the formation of malic acid, which is the 9th reaction in the presence of enzyme fumarase. This malic acid will get converted into oxaloacetic acid in the presence of enzyme malic dehydrogenase. Again, because it is dehydrogenase, so there will be removal of hydrogen ions which will be accepted by NAD+. It will get reduced into NADH plus H plus. So again there will be the recovery of oxaloacetic acid. So because 2 molecules of pyruvic acid are there, so that is why 2 times this cycle will go for 1 molecule of glucose. Now this is called as TCA cycle because 6-carbon compound, that is citric acid. TCA means tricarboxylic acid cycle. So that is the citric acid cycle. Then because Hans Krebs discovered it, so it is called as Krebs cycle. So another name is citric acid cycle or Hans Krebs cycle. So today we'll be studying only this much. Thank you.